The next organelle on the list is the Golgi apparatus. Sometimes you see it like you see in the top right, the Golgi complex or the Golgi body. Golgi is Golgi is Golgi. This is the only organelle that is capitalized, and I would use my think meat and say that's probably because it's named after somebody who discovered it. Like, let's say Mr. Golgi, because I'm sure it wasn't a woman. Not because I'm being sexist or anything, but because a lot of the people that get credited for things in science have been men, because men have been the only ones that are allowed to be scientists in the past. Anyway, that's my public service announcement for the day. The Golgi apparatus is a system of membranes very, very similar looking to the rough ER, only there are not ribosomes on the surface. But again, if you take like half-filled balloons and you squish them together, that's what the Golgi apparatus looks like. The function of the Golgi is the processing, packaging, and secreting organelle. What does secrete mean? Secrete means to release something into like the outside of your structure. So it's making something that's going to be exported outside of the cell. Now, the Golgi, again, is a system of squished sacs. So the sacs that are nearest the nucleus are going to receive vesicles, which are basically just kind of like membrane containers from the endoplasmic reticulum that contain new proteins and lipids, because remember, proteins and lipids are made on the ribosomes on the ER. And then the vesicle, or this membrane container, travels through the Golgi, transporting the substances as they go. Inside the Golgi, the proteins get the address labels that direct them to various parts of the cell. This is why I like to call the Golgi apparatus the post office of the cell, because it is going to modify the structures and basically kind of give them the instructions of where they're going to be exported to. So this is a very nice diagram that combines the nucleolus, ribosomes, ER, and Golgi all in one. So if we look at this is the nucleus, right? So ribosomes are made in the nucleolus, and you see how the rough ER is right outside. It's actually attached to one of the nuclear pores, and it's covered with ribosomes, and you've got the smooth ER here. So you've got the ribosomes, which come out of the nucleolus. They enter the ER to get modified, and they go to the smooth ER. They come out. This is a transport vesicle. Keeping in mind that when I say that they're membrane bound, the membrane that I'm referring to is exactly the same lipid bilayer that the cell membrane is made of. It's all the same structure. So a vesicle, which is my next organelle I'll talk about in the next video, it is literally just a hollow ball of lipid bilayer. Finally, the proteins come out of the ER surrounded by the vesicle. The vesicle transports them to this, the cisternae, or the flattened sacs of the Golgi apparatus. You've got, you don't have to know cis and trans because that's too complicated for now, but you see what I mean about the sacs that are nearest the nucleus receive the vesicle. The proteins are then released into the inside of the Golgi. They get their address labels, and then finally they get, they kind of blob out into a new, what's called a secretory vesicle, because we're going to secrete it outside of the cell. And because the vesicle membrane is made of the same thing as the plasma membrane, it can literally fuse or combine with the cell membrane and release the proteins to the outside of the cell. That's, in a nutshell, how protein export works.